Okay, so we just finished talking about type 1 hypersensitivity, which is the allergic response. And so we're going to talk about type 2, 3, and 4. And a lot of the examples in these are going to be have to do with diseases that cause autoimmunity. And so first we want to talk about a little bit about how autoimmunity is going to develop. And so basically, autoimmunity can be thought of as an adaptive immune response. So we're talking about memory responses that target self antigens. Okay, so there are some estimations that 1/2 of all B cell receptors that go through the bone marrow response are going to be specific for self antigens. But the B cell receptor checkpoints during development are going to prevent a majority of them being activated or released into the periphery. There are a level of normal autoimmunity that is present, and this autoimmunity does not lead to autoimmune disease. However, influences such as genes, infections over time, as well as environmental factors can lead to a breakdown of tolerance and the development of an autoimmune disease. Now, for normal levels of autoimmunity, what is meant is we do have our more innate like B cells or B1 cells. And these B1 cells are going to secrete immunoglobulin, mostly IgM. And this is going to be without T cell help. And so these B1 cells secrete natural antibodies. And these natural antibodies are going to be important for protecting against a lot of different microbes that we're exposed to that aren't really virulent. So they are going to have no help, so no hypermutation, and no memory. Therefore, these are going to have low affinity, and they're going to be able to activate complement. Now, of course, that's what we know that IgM antibodies do. A lot of times they're going to cross-react with the red blood cell AB antigens. So your A or positive, B positive, AB or O. And they're also going to respond to sugars on bacteria. They are going to be involved or important in things like transplant rejection. And they are going to also have the ability to bind with low affinity to self-antigen. And these are found in such things as lupus as anti-nuclear antibodies, for example. Okay, but in general, they are going to be IgM, which is shown here, this lovely pentamer. And the natural antibodies are going to be produced without T cell help, so they don't have the ability to become IgM, or IgG, sorry. Apoptotic cells release DNA, as well as neutrophils, they release their DNA nets, and these can induce natural antibodies against anti-nuclear antibodies against DNA, for example. But typically DNA is kept inside of a cell, so it's not going to cause too much of a problem, especially since there is no memory associated with this. And this is just showing our natural antibodies binding to your AB antigens. And so one of the reasons why you have a rejection of your transplants in the past has been because of the natural antibodies to the AB antigens, and if it were mismatched, then you would get rejection. Now, true autoimmune disease is different than having autoantibodies present because those can be not causing an actual disease. Autoimmunity is going to be driven by a memory response. So you're going to have autoreactive T and B cells. And remember that B cells need CD4 help, so you always will have a CD4 helper cell if you have B cell memory. And these are going to be through the hypersensitivities type 2 through 4. Now remember, 1 is immediate, and that's saved for the allergic response. Now, autoimmunity is typically a chronic illness. And most autoimmunity is not caused by the natural antibodies we just talked about, but by conventional T and B cells. Now, autoimmunity does require T cell help. And autoreactive T cells, how this is known is because autoreactive T cells transferred from one animal to another will actually induce the disease. T cells that overcome tolerance can induce autoimmunity. Now, T cell tolerance, we've talked about this already. And so there are many different ways of protecting your body against T cells that are potentially self-reactive. We know that a breakdown of tolerance is going to lead to autoimmunity, and this breakdown of tolerance is thought to occur over time. So you have a breakdown of central tolerance over time. Now over here, of course, we have our, in the thymus, our T cells are going to be exposed to our autoantigens or self-antigens. And then these T cells will be deleted, as shown here with these little dotted lines. So if they bind to high affinity in the medulla of the thymus, they will be deleted. Antigens such as DNA are going to be always inside of the cells, and they're going to be released in apoptotic cells or other cells. Typically, they're going to be cleared by complement and natural antibodies. So these natural antibodies that we just talked about, as well as complement, can bind to DNA and eliminate them so they don't activate the conventional B cells and T cells. We also have tissues that have privileged sites, such as the blood-brain barrier. And then we also have the ability to create an anergic T cell through the lack of co-stimulatory molecules. In addition, you have Tregs that are going to be important for suppressing potential autoreactive T cells. And then you have repeated antigen stimulation leading to activation-induced cell death. So all of these are important in tolerance. So in this case, we're hiding your antigens. In this case, we have no signal 2. And then we have Treg suppression. And they're all going to play a role and be very important. Okay, so we have genetic factors that are going to have an influence as well on autoimmunity. And so we have some rare genetic diseases such as autoimmune, polyadenocrinopathy, candidiasis, so that's the yeast candida, ectodermal dysplasia. This is a PECED. And this, they have a lack of air. Now, air is autoimmune response elements, and this is the gene that gets turned on in the medulla of the thymus to create that nice snapshot of your body. And so this is caused by gene mutations in the air gene. It's going to lead to sort of a very widespread type of autoimmunity, and it leads to the inability to control candida infections, which is going to present typically with oral thrush. Another rare is going to be a FOXP3 deficiency. Three. And FOXP3 is, of course, the transcription factor for Tregs. And so you lack Tregs, and typically it's death within a year of life, unless you have some sort of transplant. Now we have common autoimmune diseases, and these are going to be type 1 diabetes, celiac disease, and so you're going to have, because these are more common, instead of these rare mutations, you're going to have common polymorphisms, which are going to be linked to a cause of a breakdown of tolerance rather than the rare air or the rare FOXP3. So these are going to be associated with common polymorphisms. Now, for type 1 diabetes, genetic influence can be in two different ways. The first is that you can have the amount of insulin expressed in the thymus can be different depending on the genetics. And of course, the less insulin is actually produced in the thymus, the more the opportunity of an insulin-specific T cell to get released into the periphery. And of course, HLA, oops, not ILA, HLA has an influence as well. Okay, so environmental factors can also have a role, not just in genetics. So this can be by exposure to something in the environment, and it can be by UV radiation, infections. So during a UV, we're showing here a lupus patient, and they have the butterfly rash on their cheeks as well as their toes. They have some rending of their toes. And so basically in lupus patients, you have exposure to sunlight, so systemic lupus, plus UV. And what UV does is going to cross-link your DNA, and it does that to everybody, not just the patients that have lupus. And of course, we all have our repair mechanisms, and it doesn't do too much damage. It can't cause cancer, as we know, skin cancers, but it has these breaks in the DNA, and people with lupus have anti-nuclear antigens that can bind to the DNA. And then they're going to bind, they're going to induce inflammation, you're going to get leakiness of your endothelial cells, you're going to get red blood cells moving into the tissues, and that's going to lead to this rash. And so that happens with the UV. Now, with respect to infections, you have to keep in mind that during an infection, you have APCs that see danger signals, and they are going to upregulate CD80, which is going to be for signal 2. Now, you also have expression of self and pathogen peptides. And so during an infection, there is possibility that you might have a self-reactive T cell that sees a self-antigen, and because there's an infection, they also get signal 2. So therefore, and this is 1, we'll put 2, is you have cross-reaction. And this is where you have something called molecular mimicry. And this is where you have an antigen from a pathogen that looks similar to a self-antigen. An example of this is going to be acute rheumatic fever. And this is caused by strep infection. And you have a rash. And the antigens for strep the antibodies for strep cross-react with heart antigens. And so you have inflammation of the heart. And you can also have impact in the nervous system. And this is transient. So it's not a true autoimmune disease. As soon as you clear the bacteria, the cross-reactivity goes away. And there's also some data that shows a decrease in exposure to microbes. May be responsible for an increase of autoimmunity. And I say may because this isn't an absolute. I'll say contributing. So while we get cleaner, which is important because there's a lot of microbes out there that can cause disease, it might predispose us for actual autoimmunity as well. So again, we have some risk factors for autoimmunity. We have our AIR gene mutations for APECED, as well as other autoimmune diseases. We have insulin gene polymorphisms, and this is going to impact how much insulin is going to be made in the thymus. We also have MHC polymorphisms that are going to influence those more common autoimmune diseases, not the rare autoimmune diseases. We can have mutations in our mannan-binding lectin and complement. That's spelled wrong. C1 polymorphisms, which can lead to systemic lupus. And of course, another rare is FOXP3. So this is rare, and this is of course rare as well. We have we have environmental influences. Infection can give you signal 2, and you may have autoreactive T cells that are going to respond to self-antigens in the presence of signal 2. We're going to talk about medications that are going to lead to autoimmune diseases. UV light can influence the phenotype of autoimmune diseases, as well as celiac disease. We have some dietary influences there. So in general, test for an autoimmunity disease is going to be detecting autoantibodies. Now, this does not always mean that the antibodies are causing the disease. It can be caused by a T cell, but we typically don't measure T cells because they're harder to measure. We typically measure antibodies. So you have two different mechanisms, usually done by an ELISA. And again, we've talked about ELISAs already. So it's ELISA is common. So you have some antigen that's coating a well, and then you have a antibody that is tagged, and this would be a direct. So you have a fluorescent antibody. And this can be done on a tissue biopsy, for example, skin biopsy. And you can use the targeted antibody to bind to the antigen on the surface of the tissue. You can also have an indirect in which you take serum. So here, this is mostly used in tissues, and this is a patient serum antibody. And the serum antibody, of course, does not have a fluorescent label, so you have to use an indirect where you give a second antibody that has the fluorescent label. So that's all the difference is, really, is that which antibody is actually labeled. This is a primary antibody that's actually binding to the targeted antigen. So this is target antigen, and this is target antigen, of course. And so that would be the direct, or is it the second antibody that is not specifically binding to the targeted antigen, but binding to an antibody that is bound to it, and that would be indirect. Okay, so let's talk a little bit of type 1 diabetes. Type 1 diabetes is actually a type 4 hypersensitivity. And so this is going to be mediated by T cells. And they basically destroy the beta cells in your pancreas. Now, the trigger for this can be an infection. And you have basically autoantibodies that are going to be used for diagnosis, but are not really needed or the cause of the disease. So we have autoantibodies. And for type 1 diabetes, diagnosis is usually by the clinical symptoms. increase of glucose, ketones, bodies, in urine. And we also have, let me move my autoantibodies. We have autoantibodies are present, but not disease causing. Okay, so there is going to be a concordance rate associated with a lot of these autoimmune diseases. And remember, concordance rates are how likely typically a sibling or a twin will get the disease. So in type 1 diabetes, the concordance rate for identical twins is about 50%. And so this means that you're going to have an environmental influence. And it might be an infection, for example. Trigger might be an enterovirus has been suggested. Such as a Coxsackie virus. And then your HLA subtype, and it's going to be DQ2, has a 90% risk. For identical twins. So in your HLA DQ beta chain, at position 57, you're going to have an aspartic acid. Oops. And your HLA DQ2 position 57 in with the higher risk is going to have another amino acid in place of aspartic acid. And this other amino acid can be many different types. So this will increase your risk for type 1 diabetes. So what is thought is that the change of the HLA-DQ is going to alter the peptide binding, and the autoreactive T cells will not get deleted in the thymus. Oops. Oh, so this is autoreactive T cells. Okay, so this is thought to be the little pictures there are sort of just an idea about how this is actually occurs. Now, celiac disease is also going to be a type 4. So this is going to be T cell mediated. And celiac disease was once called the banana disease because physicians would give children who were suffering from celiac disease, they would take them off their diet and put them on a banana diet, and their symptoms would go away. So it was actually a Belgian who discovered that it was the gluten during a wheat shortage, because all the children who suffered from celiac disease all of a sudden recovered, And then when the wheat came back, they all got the disease again. And so he discovered, or he made the link between the bread and the wheat. He didn't really know exactly what it was causing the actual disease. And so celiac disease is a very debilitating disease because basically it converts your small intestine to a morphology similar to your large intestine, where it loses those villi that are important for absorption, and you basically are not able to absorb nutrients. And so you have an ELISA to test for a celiac disease. You use an ELISA, and this is going to be testing for an anti-transglutamase antibodies. And so you have wheat, rye, barley. They're all going to contain gluten. And then the gluten contains gliadin. And then gliadin is going to be present in the normal diet and is not normally going to be antigenic. But we have tissue transglutaminase, and this is going to be present in our normal tissue and again is not antigenic. And this tissue transglutaminase will bind to gliadin and creates glutamic acid. And then gliadin can bind to, as a glutamic acid, can bind to HLA-DQ2. And then T cells responding to this acid will help B cells responding to the tissue transglutaminase. And so it's kind of like a glue type of thing that holds it together. There is very high concordance rate. And it's going to be about 75%. Now, systemic lupus has a concordance rate of about 60%. And this is HLA-DR2. And basically, one of the bigger ones is going to make anti-nuclear cytoplasmic antibodies. And then anti-DNA. Oops. And so this is going to be a type 3 hypersensitivity. And this is going to be our immune complex. So in patients, your antibody is going to bind to release DNA. And again, this can be from an apoptotic cell. So here's our released DNA. Or it can be from those nets. So neutrophils release DNA if they're trying to counter bacteria. And they're going to form immune complexes. So here's our immune complexes. So just note that your antibodies do not bind to intracellular DNA. This is all extracellular DNA. And so the immune complexes are going to travel through the blood, and they're going to get trapped in certain locations and create aggregates. And so these aggregates are what are going to cause the disease, because aggregates anywhere are going to cause inflammation. So you have antibodies that are going to bind to free DNA, and this is going to form immune complexes. And this is going to deposit in tissues and increase inflammation. Now, lupus is, of course, systemic, and so inflammation can potentially be systemic. And systemic lupus also affects women more than men. And is thought to be related to increase in estrogen. You also have, this is sort of a timeline of development of the actual disease, so there are several steps that are thought to be important. You have, of course, inheritance of your HLA-DR2, and then you could potentially have inheritance of impaired DNA clearance, so other genes are thought to be involved. We have our mannan-binding lectin and our complement. And these will remove DNA. And so if there are mutations in them, then they can't remove the DNA. And so this could potentially lead to activation of self-reactive B and T cells. We have T cell help with low-level antibodies made against DNA. And then you can have infections that are going to increase apoptosis, increase free DNA, and add to this response. Sometimes drugs. So we can have drugs interfere with DNA clearance. This is due to inhibition of some of our methyltransferases. And this is hydrolysine and procainamide are two drugs as an example. But basically it takes time, and then there are some other mechanisms that are thought to be involved, and then you get symptoms that are going to present. Okay, so that's just the introduction to autoimmunity.